Well, good afternoon, everyone. Welcome to this latest MASTS webinar. Today we're joined by Natalie Millán, who is from the Scripps Institute of Oceanography in... she did her PhD at the Scripps Institution of Oceanography at the University of California, San Diego, but is currently an assistant professor at the University of Baja, California. Natalie is currently part of the MASTS Visiting Research Fellow program and is working in the Duncan lab at the University of Strathclyde in Glasgow. Her research encompasses aspects of marine microbiology, phylogenomics and marine natural products. And Natalie is talking with us today about assessing the biotechnological potential of the Antarctic and subarctic sediment cores as a possible resource for sustainable antibiotic drug discovery. Just a quick note with regard to questions. If you have any questions for Natalie, if you could please type these in during the webinar, and she can then address and answer them at the end. At this point, I'll now hand over to Natalie. Natalie.
Oh, thank you so much, John, for the kind introduction. And I'm really happy to be here to give this webinar. So thank you so much. And thank you to MAST, actually, for this visiting fellowship that I got to work in the University of Strathclyde. And now I can share this really interesting project that I've been working in the exploration of the sediment cores of Antarctic and subarctic as a new resource for antibiotic drug discovery. So why do we actually care to find new antibiotics? So I will start saying that antibiotic resistance it's become a major problem. It's actually estimated that more than 700,000 people worldwide die every year for infections that no longer respond to antibiotics. And it's actually estimated that the year 2050, more than 10 million people will die. So this just means that routine surgeries and minor injuries can really become life-threatening. So how can we actually stop this? How can we find new chemistry, new antibiotics? And I really want to make a parenthesis here because there's another problem related with antibiotic resistance, and it's due just in the United States, for example, a third of the antibiotic prescription aren't actually needed. And it's worse in other countries where they actually don't have any control, so there's actually no prescription. So the really problematic of this antibiotic resistance. So having that on mind, that will be something to really address for this problem. But for now, we're trying to just address the part of the exploration and find new antibiotics. So where can we find new antibiotics? What kind of environments we can explore? So since the beginning of the antibiotics we have found in different environments. Some of these are like the ocean, the bottom of the ocean, some extreme environments like deserts or other really extreme environments in the earth. And really interesting, some symbiosis with microorganisms or different organisms, marine or terrestrial organisms like this plant and interaction that can lead to very important antibiotics that we have. Another really interesting is the human microbiome. This is a new field that have been explored to find new antibiotics. And since then, there's been a lot of research going on actually besides the human microbiome, just trying to swap everything in the world to try even like dirtiest places in the world to find new antibiotic resistant genes. And there's actually a really interesting project going on right now. It's called MetaSub, like metagenomics in the subway around the world. So they actually have the major goal of find antibiotic resistant genes to trying to find new pathways that they can find and develop new drugs. So we can find new antibiotics and new drugs for different diseases. So this is being like all this exploration in different environments for that specific problem. So one really interesting environment that haven't really been explored for this purpose are the polar regions. And this environment, it's actually one of the environments has been observed to have major effects on global climate change. And specifically, the accelerated rates of warming compared to other ecosystems are way higher. So some model predicts that an increase of 4 degrees by the end of the 21st century. So this trend of increasing temperature is related, of course, of the trend in CO2 levels. So here in this graph, we can see in the x-axis the year since the beginning of 1950s to 2000s, and in the y-axis we see the CO2 concentration. So you can see the trend of being increasing with time. So this trend of temperature is related with the CO2 driven by fossil fuel burning. So we really want to have a better understanding of this. So these changes are actually causing major problems. And some of these major problems in the water systems through more droughts in the different cities, more flooding, and of course the melting of the glaciers impacting the freshwater ecosystems. So not just the habitats for polar bears, but also affecting all the other organisms that are living in this environment. So the polar regions are really affected by this climate change. So there's been a lot of research involved in these effects in other organisms, and here I just shared with you some of these changes in invertebrate communities in the polar regions, like how these organisms respond to these changes. Another research going on are about bacterial and archaeal communities. And this last one about the microbial diversity and functional capacity in polar soils are really interesting research going on about the response of climate change and why is this communities, invertebrates or microorganisms important in the polar regions. Well, they actually are really important and a major component of the total biomass and mediate the cycling biogeochemical cycles like nitrogen, carbon, and another major elements. So some of these major groups of bacteria are Proteobacteria, Acidobacteria, and Actinobacteria. So actually simulated experiments have shown that microbial community changes in response to climate change. So this changing in these polar regions, changing in these microorganisms are really important, and we need to know the consequences. So besides the importance in the community of the polar regions, these organisms like invertebrates and bacteria have another importance, and that's because they produce a lot of natural products that we use for our own benefit as for diseases or antibiotics. And here's just one graph of a distribution of marine products by different phylum. So you can see that the first graphs are microorganisms, and then the biggest are sponges and cnidarians. There are some invertebrates, and you have on the y-axis the number of compounds that they actually discover by year. So besides being really important in polar regions, they actually produce a lot of these natural products that we use. So sponges and cnidarians and bacteria are this really important group that can help us to find new drugs. And I will focus my presentation in bacteria. And why bacteria? I think most of the people that think about bacteria will think that bacteria can just cause me a disease. Here's just an overview of bacterial infections. You have eye infections, you have gastritis, that's really common. You have skin infections by Staphylococcus aureus, that's actually bacteria that we have around all our skin. Pneumonia that can lead to really big problems. So most of the people who think about bacteria will think, oh, it will kill me. But actually it's being bacteria, the ones that are giving us these antibiotics. And bacteria is one of the most genetically diverse and ubiquitous domains of life. Here it's a view of a new view of the tree of life. You have, you can see that bacteria is one of the most diverse. And if we think about the natural products that we have explored from this domain, we have that more than 50,000 natural products come from bacteria, and just an estimation that 80,000 have antibiotic and antitumoral properties. So they are giving us a lot of this potential. So we can focus on a lot of different groups within the domain of bacteria, but I will focus my attention on one specific group, and that's because it's been well known that if you find a new bacterial species, that's been really, really important for research institutes and universities to really try to find new antibiotics, try to lead this research to try to find new compounds and that new species will lead to new antibiotics. So that's the main goal. So like I said, I will focus my attention into trying to find new antibiotics and a specific one order of this domain, that is the order Actinomycetes. So this order of Actinomycetes within the domain bacteria is a gram-positive bacteria that has a high GC content. It's a filamentous growth, and it actually produce and form spores. So you can see in the picture the branching, that's the way that how they grow, and then the spores within a sediment that's in that picture, and that's the typical morphology of the actinomycete bacteria. So this group of bacteria produce a lot of secondary metabolites, and actually it's known that 70% of the microbial antibiotics discovered today comes from this group. And just to mention some of very common antibiotics that we use, we have erythromycin, tetracycline, chloramphenicol, rifamycin. So all this list of antibiotics that I just mentioned are coming from natural products produced from this order of Actinobacteria. Specifically, one group of genus of this order is Streptomyces. Streptomyces, from that 70% of antibiotics coming from Actinobacteria, 80% of these natural products that has biological activity come from this genus, Streptomyces. So you can see some of the morphology of this Streptomyces strains. And another really important research that's been going on is to sequence the DNA of this bacteria to know how much of their genome is being for just the production of secondary metabolites that can lead to new antibiotics or new drugs. So there's a lot of research going on right now, and it is known that around 6 to 10, 11% of their genome is dedicated for the production of secondary metabolites. So that's a really important information to know when you're like trying to find new species, to know how much of the genome can help me to find new drugs. Besides the Streptomyces, there's been another genus of Actinobacteria that people have been doing more research. And here in this slide, I show, it's a really busy diagram, but it's a project that just been published last year by Shorn et al. In UCSD in San Diego. And they found that besides the Streptomyces, there are a lot of different genera of Actinobacteria that can actually produce a lot of different natural products. So here, if we see the graph, we see that the half of the plot, it's a different genus. So we have Actinomadura, Pseudonocardia, Nocardia, Rhodococcus, some of them. And then the other half of the figure, it just comes the potential of clusters that it can produce different secondary metabolites. So we have NRPS, PKS, siderophores, terpenes, so the different chemistry that they can produce. So besides the Streptomyces, there are a lot of different genera that can actually lead us to find new compounds. So knowing that part, here in the Duncan lab in Strathclyde, they actually did a first project trying to find new secondary metabolites from isolates from the Antarctic. So they actually had samples from Scottish and Antarctic sediments, they found the genera Streptomyces, and they actually found that some extracts of this bacteria have activity against colon cancer cells. They did a lot of the chemistry to know if what they produce, this fermentation that they do to grow them and then extract them, to know the chemistry. So they use this program, this approach that is called molecular networking. And here's a part of this project that they did. And I just want you to focus that you can find a lot of information in this graph. But in blue, you see the compounds that have been isolated from Antarctic extractions of the bacteria they have found from there, and in pink, the ones from the Scottish sediments. So the numbers in these nodes just represent the mass of the compound. So here you have this compound that the mass is 650, and then that will represent that node. And then you can have standards and trying to know what kind of compounds they are, but the more interesting will be something that haven't been seen before. So that means that you can have new chemistry in your extracts. So that will be like a new mass. And then you can see how many of the Scottish sediments are similar to the one in the Antarctic. So actually doing this comparison, they found that 40% of the chemistry found in the Antarctic is very specific for that location, and the other 42% are from Scottish sediments. So it's actually really interesting because that means that bacteria from different locations are actually producing different chemistry. So the opportunity to find new molecules, new chemistry is higher if you find and compare different locations. So with that in mind, we actually applied for this fellowship, this MASTS fellowship. So I came here at University of Strathclyde with Kate Duncan, the professor at this university. And thanks, I would like to thank again MASTS for this visiting fellowship that I got. And also the Scottish Association for Marine Science in Oban, because we use their collection of marine sediment cores to start this project. So the major goals of this project was to do the culture-dependent diversity, just trying to find new actinobacteria, and also do the genomic studies to sequence some of the most important strains that we have isolated. The second part of the project will be culture-independent studies, so trying to do some metagenomic approach, try to isolate the DNA from the sediment and sequence them to know the total diversity of bacteria that we can find, besides the one that we can culture in media. And the third part will be do the chemistry, especially this molecular networking, so we can find the different metabolites that we can compare between the Arctic and the Antarctic sediments. The major goal of this will be try to find new diversity, new biosynthetic potential, and of course biotechnological applications to try to find specifically new antibiotics. So how did I start with this project? Like I said, we went to the Scottish Association for Marine Science in Oban, in Scotland. I had the opportunity to go to the collection that they have. So they have the sediment cores, and I went and took 12 sediment cores in total, 6 from the Antarctic and 6 Arctic cores. And this one comes from different expeditions. So here's just some of this data. The length of these cores are different. There were some from 16 centimeters, and one of the biggest ones that we took were like 87 centimeters. And really interesting part of this collection is like some of the sediment cores that they have, they actually come from depth of more than 3,000 meters. So it's a really unique opportunity to be able to explore the sediment cores. And like I said before, they come from different expeditions, 2002, 2005, and 2010. So once we got there, we actually saw where the sediment cores were coming. So like I said, we had 6 from the Arctic and 6 from the Antarctic. And I was able to collect different parts, different sections of the sediment core. So here in this picture, you can see that I tried to take sediment from the upper, the middle, and the bottom section of the sediment core. And here, this was for a couple of reasons. We wanted to compare the diversity of these different zones of the sediment core and to try to have a better explanation of the different diversity, and also to try to correlate to different parameters in these sediment cores. Here's another picture of one of the sediment cores that we took, and here's just like a zoom in of like how the sediment looks. So like I said, we have 12 sediment cores and then 3 different sections. So we collect everything, the samples that we needed, and we put the sediment cores back into the collection because that was part of what we decided to do. So we have it there. And then we just went back to Glasgow, where I just put them in the freezer, and then to start the second part that's trying to grow some bacteria from the sediment. So first thing I did was to dry a little bit of that sediment into the hood. This is for a couple of reasons. We want to dry the sediment because they are, what we're focusing are in Actinobacteria, so they actually produce spores. So drying in under the hood will kill some of this gram-negative bacteria that we are not really looking for. So we want to dry the sediments so the spores will resist this drying process. Then after they're dry, we can stamp them in different media. So I used 3 different media, and then I used the method of stamping the sediment within the plate. So you do kind of a dilution. You can see in the picture there is a dilution of the sediment that we stamp, and then these bacteria take a little bit time to grow. So we just let them to grow in our bench. It was actually a room temperature. We didn't really try different temperatures. It was the thing that we had here at the university. Here's just like close-up of how this plate looks with the sediment. And here's where the patient part starts, right? Because we just want to check the plates once in a while to see if we can see isolate some colonies, so we can like pick them and then pass them again until we are sure they're like pure cultures. So it's a really cool part because you can see a lot of diversity, different colors. Here's a picture of some of this bacteria that I have isolated. So you see on the top like some yellow color, some white, some orange. And so then you're passing them at least 3 times into a new plate to make sure these plates are pure cultures. So then we can do DNA extraction, get their DNA to send to sequence and know what kind of bacteria they represent. So here's just some cool pictures. Well, I really love bacteria, so I'm like really looking into the morphology. And this is one of the bacteria I have isolated. They're like orange with this white, it looks like a white powder, but we believe that white powder are the spores that actually produce. Here's another picture of that. So this is more like yellowish color in contrast with the other ones on top. They're more orange. This is another different morphology that we found in the isolation, and this is more common ones of bacteria that you find when you're like picking bacteria, right? So more slimy looking ones than the other ones that you see in this slide. So like I said, I grew this bacteria. Something that we do in a lab after you have cultured this bacteria is to start a culture collection. So we actually grow them in liquid culture, make sure they're pure, and then we cryopreserve -80. That's the way, if you want to grow them again, we have the vial and we can do more analysis with them. So the idea is to do the DNA extraction and do the whole genome sequencing. But for now, we just sequence the 16S. So the 16S is a gene that is really conserved in bacteria, and it will tell you what it is, like what species it is, what genus, what kind of, in all this tree of life of bacteria, what order they actually clade. So this is a 16S phylogenetic tree. In blue, you have the Antarctic samples isolated, and in orange, the Arctic ones. you pay attention to this tree, you see that some of the clades are really just representatives of the Antarctic ones, and some of them are in the both polar regions are represented in some of these clades. And one of the different isolates that we have isolated represent different orders of the Actinobacteria. So just to show you, so far we have 45 isolated strains, and they represent different genera. We have Pseudonocardia, Rhodococcus, Microbacterium, Pseudomonas, and also some Bacillus, some Bacillus strains. So to try to clade them in different phylum, we have the phylum Actinobacteria. That was some of the bacteria we are going to find new chemistry, but we also found some representatives of the phylum Proteobacteria. Like I mentioned, they're really common in the polar soils, and we found them, and they're really cool morphology too. So I isolate them for that reason too, and some of them are known to produce secondary metabolites. So the potential of finding new chemistry will be higher. And also we found some of the phylum Firmicutes. So I can go and really what I'm doing right now is exploring any of these clades to see the similarity with strains that have been isolated before. So I will focus on the phylum Actinobacteria in one specific clade, and this is the Pseudonocardia clade. I'm focusing on this one because of really important features that I found. First, the morphology that I have never seen before. They actually clump a lot, and it's kind of hard to work with these strains because once you're trying to pass them to new media, they kind of collapse. So it's been like really interesting morphology-wise. So just trying to explore more into that, I think that will be like another thing that we can explore, another field of research to explore more of the basic biology of these strains. But focusing on the 16S tree, you found that the closest bacteria based on 16S is Pseudonocardia petroliophila. And you see that the strains isolated from the Antarctic are actually sister taxa, but they're not 100% similar to this type strains that have been described. They're actually between 98% similar. And then you see that these strains are coming from an expedition from March of 2002. And this clade is actually coming from an expedition in September. This B just means the section of the sediment cores where it was like isolated. So all this clade has been isolated from the bottom part of this core. And this one has been isolated from the three different sections, like the bottom, the middle part, and the upper part of the sediment cores. So it's been interesting. We like found it, but even they come from two different sediment cores and they don't actually like overlap. So that's something like pretty cool to see in this phylogenetic tree. This is the map where they actually have been isolated. So they're really similar. We're having took the sediment core. And if you want to see some difference besides the 16S, of course, because if they were 100% similar, they will play together. So you can see now that they have some difference based on 16S, but also the morphology. Actually, the color. This one's isolated in September. The I isolated are a little bit more orange looking than the other ones. They're a little bit more yellowish. I know it's a little bit hard to see, but I hope you can actually see the differences in the morphology. So these are some of the strains that we're really, really interested to sequence to see more about their potential. So that's part of the project that is going on right now. So what is next and what I'm doing like in this at this moment? I'm finishing actually the culture collection of the strains isolated from this 12 sediment cores, and now I'm trying to choose which ones are we going to send to sequence the whole genome. So that will be the second part. And also right now I'm actually growing most of them in liquid culture to know more about their chemistry. With that, we can do some molecular networking analysis and we can actually see the difference in chemistry that they can produce. And that will be the most important part of the project. That was the culture-dependent part. The second part that's also, I've been working on that since the beginning, is trying to do this metagenomic studies of the sediment cores. I've been trying to isolate the DNA from the sediments, from the core. I've been having a little bit of problems to really low yields of DNA from the sediment cores. I actually just went to a symposium last week, and I talked to some people about the better yields of DNA that we can isolate. So that was a really useful information I found last week in this symposium in Warwick University. And the last part of this project that it will be more in detail about the exploration of this bacteria. We'll be trying to link the biochemical parameters that they have of the sediment cores, and trying to find a little bit more of the ecological function of this bacteria and the chemistry that they produce. So that will be something that it can lead to future projects. But for now, we have had a really set of cryobiotes and strains that we can actually look for new chemistry. So that will be the next steps to do. So that's what I'm right now. So with that, I just really want to acknowledge, especially the people in the Duncan lab, especially Professor Kate Duncan, Jordan master students. They've been helping me with the isolation. And of course, the other two professors, Paul Huskisson, Paul Heron, for the be able to use the resources in the lab. Once again, I want to really thank MASTS for the visiting fellowship that I got to work in this really interesting project, and SAMS, of course, for the be able to go there and take some of the samples from the sediment cores. And of course, I want to acknowledge my university, University of Baja California, that gave me the opportunity to also be part of this collaboration with the University of Strathclyde and be able to keep doing this really interesting research. So with that, I would really like to thanks again for listening, and I'm really happy to take any questions.
Thank you, Natalie. That was really, really interesting. Just check. I'm not certain we have any questions at the moment. If anyone does have any questions, if you could please type those in. I've got a quick question. I'm just wondering, in terms of selecting the Arctic and Antarctic, you said that they've not really been explored. But I was wondering whether sort of strains from a cold environment, there might be a higher probability of them producing inhibitory metabolites. As they're so slow growing, they'd rely more heavily on those than, for example, sort of warmer environment strains, which would rely on sort of growth rate to outcompete other strains.
Yeah, that's a really interesting point. And right now we're just like growing them in like room temperature. So something that we're trying to do is grow these strains again in like lower temperatures. So because they come from more cold environments, right? So that's been what we're trying to do right now, to keep exploring more diversity.
Okay. Have you maybe not thought of doing sort of direct competitive culturing? Just picking some common strains and then sort of co-culturing with the strains you found to see if there is any sort of observable inhibition.
Inhibition? Yeah. So the reason we haven't started with that is because we wanted to finish to have our like whole culture collection. So right now we just wanted to make sure that we have the strains cryopreserved, but that's the next step, because now we can do some easy assays to try to see this inhibition with like the most common ones. And they've been doing this kind of assays here. That's the reason we actually focus our attention on antibiotics, because they do a lot of this overlay assays with different bacteria, so we can have a really easy way to see this inhibition between the bacteria we have isolated and the most common ones.
Yeah, I'd be really interested. That's what I spent most of my PhD doing, looking for competitive interaction between Vibrio species. So
Oh, cool.
I'll just double check again. I don't think any of our attendees have any questions. No, so I think at that point I'll just say thanks again, and yeah, very interesting talk.
No, thank you. Thank you so much for the invitation, and yeah, it was a really interesting experience. Thank you.